So Jennifer's talk is Benthic Coral Reef Community Dynamics, Lessons Learned from the Central Pacific.
Thank you. Is the mic working? Yeah. Okay, great. Well, thank you all for coming, and I'd like to thank Mark for inviting me here today to talk to you guys about some of the work that I and members of my lab have been conducting over the last several years. I'd like to thank the Teasleys also for giving us the opportunity to get together such a great group of people to talk about such important issues related to coral-algal interactions and benthic community dynamics on coral reefs. So my talk today is going to be kind of a historical overview of my research as it relates to benthic coral reef community dynamics, focusing specifically on lessons that I've learned from the Central Pacific. So for most of us who have, let's see, where do I can just use the arrow there. So for most of us who have been fortunate enough to dive or snorkel on a coral reef, we may be familiar with landscapes such as this, especially for those of us who have been lucky enough to go to far remote places where people have had few to little impacts. And what we notice is the beautiful corals, the large architecturally structure-forming organisms that build the complex habitats that reefs are known for. We see these corals, the reef-building corals, growing up towards the sunlight, depositing calcium carbonate, and actually building the habitat that so many species are dependent upon. These corals are active, corals and coral reef habitats in a healthy state are actively accreting, they're laying down calcium carbonate, they're building reefs that actually humans depend on a lot of the ecosystem goods and services that these reefs provide, one of them being their ability to break up wave energy and disturbance, preventing erosion from happening in coastal ecosystems. Coral reefs also provide huge economic value in terms of fisheries and tourism. So we see the corals dominating these landscapes. Again, these are in places that are relatively pristine and untouched with little human impact. The corals themselves are kind of the charismatic megafauna of the benthos in these images. They're what we see are the large, beautiful, and again, architecturally ecosystem engineers in these images. But on any given reef, corals are not the only organisms present. In fact, we have many other important organisms that occupy a reef, that are present in a reef community. We have various species of macroalgae. We have calcified macroalgae such as halimeda and a variety of fleshy macroalgae, all which have different ecological roles on a coral reef. We have the crustose corallid algae. These are pink, red algae within the certain order, the corallinales. These algae are also active reef builders. They lay down calcium carbonate. They are also known to facilitate coral recruitment. And so they're generally a good indicator of a healthy reef ecosystem. We also have the more diminutive and less conspicuous algal terps. These are a small consortium of small filamentous algae, cyanobacteria, and other groups of little small seaweeds. While they're less conspicuous, they often occupy a large component of the benthos in any given reef community. And then we have a variety of invertebrate species, and all of these organisms together make up the reef community that we see. So while many of us that would snorkel on a reef would still tend to notice the brightly colored charismatic corals, I've spent much of my career focused on the smaller scale interactions that happen on these reefs, specifically looking at interactions between corals and their benthic competitors. On a coral reef, competition for space is incredibly fierce. You rarely find any open space on the benthos, and so these organisms have adapted a number of ways of competing with each other to occupy or overtake that limiting space. In this case, you see a coral in which appears to be an actively overgrowing interaction where it's actively killing and overgrowing the crustose coral and algae. This is, of course, just a picture, and we don't know what's happening over time, but that's what the image suggests. We see other cases where crustose coral and algae are actively growing over coral. We see other more benign interactions where there's no clear winner. The CCA may be gaining here, the coral may be gaining here. And then we see a whole variety of other interactions between corals and algae on coral reefs. And for any of you who spend a lot of time with your head down on the bottom looking closely at interactions, you see lots of interesting things that allow you to develop all kinds of hypotheses to test how things like these little filamentous turf algae are able to invade live coral tissue in many cases. So on any given reef, There are a number of factors that affect the composition of the benthos, and then there are a variety of mechanisms by which corals and algae and other organisms on the reef benthos interact. And it's the outcome of these two things that ultimately leads to the structure, the landscape structure of a given reef at any point in time. So that's essentially the outline of my talk. I'm going to focus first on looking at some of the factors that affect the structure of benthic coral reef communities. Then I'm going to focus on looking at some of the mechanisms by which corals and algae interact, and then ultimately how these things lead to the structure of reef landscapes that we see today. So first, I'm going to focus on a study I did as a PhD student looking at the effects of overfishing and nutrient pollution on benthic community development, and look at the possible role of herbivores as a tool for reef restoration. I'm going to look at coral-algal interactions and introduce the potential role that microbes may play in coral-algal interactions, and then we're going to evaluate the structure of reef communities on islands across the Central Pacific and look for evidence of human impacts. So starting with number one, in addition to all of the global anthropogenic stressors that are happening around the world, things such as global warming, ocean acidification, which may cause direct losses in coral cover, many local factors, and primarily things that Nancy and Pete have both talked about, that is the role of herbivores and the role of inorganic nutrients. These local factors are believed to be some of the primary drivers of reef ecosystem structure at a local scale. So on a healthy reef, we have a lot of herbivores that are consuming algal abundant or consuming algae. We have low nutrient concentrations, which keep the algal growth rates at low levels, and this allows corals to be competitively dominant in these cases. So with humans, we overfish the herbivores, we add nutrients, and both of those factors can either alone or together increase potentially algal growth rates, potentially algal abundance, and allow the algae to then become competitively dominant over the coral. So let's test this. I wanted to determine what the individual and combined effects of nutrient enrichment and herbivore removal were on benthic community development. I wanted to do this in a longish-term experiment, greater than a year, and I conducted this study in a coral-dominated habitat with high herbivore biomass and low nutrients. The motivation for this study was, at the time, very few studies had actually looked at the combined effects of these two factors, and those that had been done largely in the Caribbean in places where macroalgae were dominant. They weren't done in a coral-dominated habitat, and most of them were relatively short-term, and so I thought that by doing this for longer-term in a coral-dominated habitat, we may actually get a better understanding of how we would expect a coral-dominated habitat to shift when we add nutrients or we remove herbivores. So I did an experiment on the Big Island of Hawaii, on the Kona coast, 10 meters depth, again, coral-dominated habitat. You can see lots of Crites compressa and Crites lobata, the reef-building corals in the background. I removed herbivores by adding cages. I added nutrients by adding these little fertilizer diffusion pots. And I measured the development of benthic communities on these little settlement tiles over time for about a year. So I measured the settlement of all the species that arrived on those tiles pretty much monthly and quantified who settled on these tiles in these different treatments. So what did I find? In the control treatment, where herbivores were present and there were no nutrients, I had highly grazed settlement tiles even after a year. We see some crustous coral and algae, these pink things, and little bits of highly grazed turf algae. If we added nutrients, we seem to have gotten an enhancement of the crustous coral and algae, or at least there was a greater abundance of those on the nutrient enrichment treatment. And then when we removed herbivores, we got an abundance of both fleshy turf and macroalgae. So turf algae were dominant in both of these treatments, but when we added nutrients in addition to removing herbivores, we started seeing some of these large macroalgae showing up. So we're seeing essentially different communities developing in each of these treatments over time. We can look at the actual data. So the triangles are the herbivore exclusion treatments, and we can see turf algae dominating in those two herbivore removal treatments. When we added nutrients in this treatment, we see we got an increase in macroalgal cover. And then the two treatments where herbivores were present, we got an increase in the abundance of crustous coral and algae. Remember, these again are reef builders. They can facilitate coral settlement. So we're seeing an increase in the abundance of these reef builders. And interestingly, we only saw corals arriving in the treatments where herbivores were present, so where we had the crustose coralline algae, where the turf algae were not dominating. So we see herbivores seem to be facilitating CCA abundance as well as coral settlement. We can also look at this from more of a community perspective and say, how are these communities as a whole changing? We'll do some multivariate analyses, so we can look at our species by sample matrix, calculate an index of similarity, and then we can rank those similarities and plot each of the treatment by time combination in a two-dimensional plot. So points that are closer together in ordination space essentially mean the communities are more similar to one another. And then we can do some basic hypothesis testing to ask whether the communities within a treatment are more similar to each other than they are to other treatments. So this shows some multidimensional scaling plots over time, this being the last point, this being the first. And we see initially the communities are all kind of scattered. There's a lot of variability and no clear clustering among treatments. But as we move through time, we see the communities in the individual treatments are becoming unique, suggesting that each community in each of those treatments is a different type of community. We can look at this in successional trajectory space. So again, starting in the beginning, we see all the communities are relatively similar to one another, and over time they all diverge in unique ordination space. So the communities are, in fact, developing uniquely over time. And we can look at the statistics for this. So we see significant treatment effects for essentially all of the variables in this model. But the interesting thing that I found was these time by treatment interactions, suggesting that the communities in these treatments were becoming different at different points in time. With the herbivore effects, after 1 month, there are already differences. So we had different algal communities in the cage treatments than we had in the uncaged treatments. But the fertilization effects took over 4 months to become significant. Well, why is this important? A number of other studies have been conducted since the time that I did this and even before, and most of them found little to no effective nutrient enrichment on benthic community structure. But if you look at the time that these studies were conducted, most of them are relatively short term. And in fact, the only study, which was, again, one of our case students, Darren Benthic, conducted a study for 8 to 10 months, and he actually found really similar results to those presented here. So this suggests that there may be some important time lag in the response of these benthic communities to nutrient enrichment, especially when we start from a bare substrate. So it may take a while for the species to settle that are actually going to respond to nutrient enrichment. Okay, so we've seen that both of these factors can affect benthic community structure. We see herbivores are limiting the abundance of those fleshy turf and macroalgal species, and herbivores are also enhancing the abundance of the reef builders, the corals and the crustose coralline algae. Nutrient enrichment does also have effects, but it depends largely on whether herbivores are around or not as to what kinds of effects they're going to have. We see this timeline in response to nutrients, and ultimately the results of any of these experiments are really going to be context dependent. It depends on the type of community that you're conducting these experiments in. If you're working on a macroalgal dominated reef, you wouldn't expect your control treatments ever to be dominated by coral. If you're working in a coral-dominated reef, perhaps you have different expectations. So the results suggest that herbivores are needed to support a calcifying reef-building community, and that nutrient effects will largely depend on the herbivore population. So we were then interested in looking at whether these benthic community trajectories, the communities that develop in these different treatments, could be reversed. And so we did a simple experiment by basically just removing the cages on our herbivore exclusion plots and looking to see if the benthic communities changed over time. And so remember, this is what the plots looked like before we removed the cages. These are the two herbivore treatments, the herbivore exclusion treatments, so this is what we're going to be looking to see changes. And basically, when we removed the cages, the herbivores were essentially able to consume all of the fleshy turf algae. This isn't really surprising or that interesting. If you put a little plate of luxuriant algal turf out on a reef where there's a bunch of hungry mouths swimming around, you'd expect them to eat it. But the interesting thing was that on our nutrient-enriched and herbivore-excluded plots, we saw some lasting effects of nutrient enrichment. Largely, we had some encrusting fleshy macroalgae that did not seem to be consumed, and this could be due perhaps chemical defenses that the algae may have had. And so we can look at this in the context of our successional trajectories. Again, this is before we remove the cages. The two triangle plot figures are the herbivore exclusion treatments. So we remove the cages. We essentially see that the plots that just had cages, we removed the cages, they became not different from the controls. The herbivores were essentially able to remove all of that previous treatment effect. However, in the herbivore removal and nutrient-enriched plots, we removed the herbivores, or we headed the herbivores back, and they became not different from the fertilized treatment. So it suggests that there's perhaps some lasting effects of nutrient enrichment on these plots. Again, this is a small-scale experiment, and it's hard to scale up the results from these to any kind of larger landscape-level context. But it suggests that herbivore restoration may be able to reverse community trajectories, and that might be most effective in the absence of other disturbances such as nutrient enrichment. Obviously, experiments need to be conducted on relevant spatial scales. We need more large-scale, more kind of ecosystem or community-level data, natural experiments to really get at some of these questions, but I still thought these results are still interesting. So some of the relevant questions that remain are how many and what type of herbivores are needed to control a given algal community. And Pete's work, shows a lot of great data on looking at this from a modeling perspective and giving us some context as to where to go and where to start testing hypotheses. I have several students, one, Emily Kelly, which is working on the island of Maui at one of the world's first herbivore-specific protected areas. So they protected this reef against all take of herbivores with the specific goal of trying to reduce the abundance of this the macroalgal cover that had been increasing there over the last several decades. Emily's been working there since prior to the establishment of this protected area and is trying to work out how many and what type of herbivores are needed to balance the algal production budget. So it's basically like you have a certain amount of algae that are growing at this rate, you have a certain herbivore population, herbivores are eating X amount of algae at this rate, and how can we basically balance that budget? This is an ongoing project, and so I don't have any data to present on that. Another one of my students is addressing this from the other herbivore side of the coin, basically looking at urchins and asking the question, are all urchins created equally? Do they all serve the same ecological roles on a coral reef? Typically, urchins are just believed to be macroalgal grazers, but do they actually eat different things? Is there functional redundancy in sea urchin species? And in the Pacific, we do have a lot of different urchin species. So the results so far suggest that each urchin does actually have unique diets and, more specifically, really different metabolic rates and food demands. And then lastly, in relation to the role of herbivores on reefs, I have a master's student who spent the better part of the last 3 years, probably should be a PhD at this point, compiling data looking at conducting a global synthesis on coral reef herbivorous fish populations. So he's compiled data from the peer-reviewed literature and from academic and governmental monitoring programs to determine what the average coral reef herbivore population is. Is there evidence that herbivorous fishes have declined on fish versus unfished reefs? Not really rocket science, but there's still, aside from small regional studies, there hasn't been kind of a global analysis of these questions. And he'll be presenting these data at ICRS, so I'm not going to focus on them, but just to show you a brief glimpse across many different archipelagos around the world. Black bars are unfished areas, red bars are fish areas, and you can see there are a lot more bigger black bars. And if you look at the data overall, we see that in fished areas, there's pretty much a threefold decline in total herbivore biomass, so about 20 grams per meter squared, versus on unfished reefs around 55 or 60 grams per meter squared. So this is, again, another work in progress, and I look forward to chatting with you guys about these data. So now that we've seen that some factors, primarily nutrients and herbivores, can actually affect the players that are present on a given reef, let's move into looking at how those players actually interact and compete with one another. So back in the early 2000s, Laurie McCook and some of his students wrote a seminal review on coral algal interactions, and they summarize that the mechanisms by which coral and algae interact include overgrowth, shading, abrasion, chemical allelopathy, preemption of space, and epithelial sloughing. At the time, there was limited evidence for many of these different competitive interactions, and so we wondered perhaps there are some other mechanisms that haven't yet been included in these analyses. So our work was initially inspired by a team of scientists that were working on the effects of inorganic nutrient enrichment on coral health. They basically added a bunch of nitrogen and phosphorus to water with corals and looked at mortality. And they found very low levels of mortality with nitrogen and phosphorus enrichment, but they also added organic carbon to different forms of sugars, and they started seeing large amounts of coral mortality. This was repeated in another experiment by Neil and Kuntz and originally by Davy Klein. So coral mortality seems to be correlated with addition of sugars. So why would this be happening? It turns out that when you add sugars to this water in the corals in particular, you get a huge increase in microbial production. And so this suggests that somehow the microbes are, well, not surprisingly, feeding on DOC, increasing their metabolic rates, perhaps decreasing the amount of oxygen in the water and causing coral mortality. So where would DOC come from on a coral reef? We took a step back and looked at the literature around the world across all different types of ecosystems, and we found out that pretty much all primary producers, whether it be trees, shrubs, grasses on land, release some proportion of their daily fixed carbon. They can't actually assimilate it all into tissue, and so they leak out these sugars, in the case of trees, and through their roots. Phytoplankton do it, macroalgae do it, so why wouldn't coral reef algae do it? So we hypothesized, basically after, fortunately or unfortunately, being stuck on a ship with several microbiologists for over a month, Forrest Rower in particular, he decided to make my life much more difficult and complex by forcing me to study little things that I couldn't see. And surprisingly, Forrest has presently been taken down by microbes because he's at home sick. But we basically hypothesized that algae could release something, a compound, dissolved organic carbon, that could fuel bacterial activity and cause coral mortality. We tested this in a series of small-scale experiments. Subsequently, we've looked at the response of coral larvae to algal-released DOC, and we've also focused on quantifying the release rates of DOC and subsequent microbial production on several different species of algae. So basic methods for the original experiment were to grow corals and algae in these little chambers that were separated by an anodisc 0.2 micron filter, which only allowed dissolved compounds to pass. It didn't allow bacteria or viruses to pass. They were grown in a water bath. They had aeration. Water was changed every day. And we assessed coral health and physiology over time by measuring fluorescence and dissolved oxygen concentration. So can algae release compounds that cause coral mortality? This was essentially the results of the first experiment. The corals on the top were the controls that had no algae adjacent to them. The corals on the bottom were the ones that were grown in those chambers near algae. Seemed to be shocking results, and I didn't really want to believe Forrest at the time, but looking at the physiological data, we also see that the corals that were adjacent to algae had very low photosynthetic yield. They also had hypoxic conditions occurring on their surface, whereas the algae were producing oxygen, and the corals, in the absence of algae, were also producing oxygen as they showed their photosynthetic organisms. So it appears that something about being next to an alga where there are possibly dissolved compounds coming through is causing this hypoxic condition on the coral surface. So we wanted to see if this was a microbially mediated interaction, so we added antibiotics and repeated those experiments, and basically found, again on the top, our corals now adjacent to algae but with antibiotics, and down here we have corals adjacent to algae without antibiotics, and you can see these corals were all heavily bleached and showing signs of serious discomfort, whereas the corals on the top appear to be still healthy. These results were again corroborated with physiological data where we have severe reductions in photosynthetic efficiency on the corals next to algae without antibiotics. Also similar results with the dissolved oxygen. So again, the dissolved oxygen on the coral surfaces near algae without antibiotics were again hypoxic. So this suggests that algae are actually releasing compound that is causing coral mortality, and that mortality can be prevented with the addition of antibiotics. So this suggests that bacteria are doing something, they're responding to those algal exudates and causing hypoxic conditions on the coral surface, likely due to respiration, and thus causing this mortality. So we wanted to see how common this was. We assessed several coral algal species pairs, and this just shows several of the coral genera that we examined, several of the different algal genera and or functional groups that we examined. And we see that there's a lot of variability from coral to coral. Some corals died regardless of what we did. The croppers are notoriously fragile. Other corals had little to no effect, and others were more susceptible to this algal damage. But in general, if we looked at the response of corals with and without the addition of antibiotics, we see that there's the majority of corals that had no change in tissue and pigmentation or physiology were the ones that had antibiotics added. And we saw a large proportion of corals that suffered some sort of mortality in the absence of antibiotics. So again, this suggests that the mechanism appears to be prevalent across many different taxa, but that it's definitely species specific. So now, what about, we've seen that adult corals or coral fragments can respond, or responding to these algal exudates. What about coral larvae? So we've been collaborating with Dr. Mark Vermey, otherwise known as the coral whisperer. This Mark can grow coral larvae unlike anybody that I know. And so Mark wanted to test the hypothesis that coral larvae also are responding to algal exudates. So he took planulae and exposed them to, this is a Ulva fasciata exudate. We have these two treatments do not have algae exudates present. These two do, so we had the greatest survivorship of coral larvae occurring in the absence of algae. He had the least survivorship of coral larvae happening with algae and no antibiotics. You could add antibiotics and get a slight increase in coral survivorship, in the larval survivorship. So this suggests that there's something microbially going on here. He also tested this with several different algal genera and found, again, some species-specific responses, suggesting that larvae are more or less susceptible to different types of algal exudates. And that, yeah, algal-induced larval mortality differs among algal taxa. Mark repeated these experiments. The first ones were done in Hawaii. He repeated these experiments in Curacao with several different types of seaweeds. Again, Acanthoptera, a red alga, Dictyota, a brown, Halimeda, a green, Lobophora, and two types of turf algae. And he found, again, that the results were variable by taxa. Some species, like Halimeda, had little to no effect on larval survivorship, but the degree of mortality really depended on the types of algae that were present. The mortality could be reduced to some degree with the addition of antibiotics in some cases. In the case of turf algae, we had just significant reductions in survivorship. Turf algae appeared to have some pretty detrimental effects on these coral larvae. So we've seen that coral larvae can also be susceptible, and again, there seems to be some species specificity. Things like Halimeda having little to no effect, and other algae like these consortium of mixed turf algae having significantly large effects. So why, despite the fact that we've published a few papers on this, several of our colleagues still, and arguably so, didn't believe that algae actually released DOC. So we kind of went back and reinvented the wheel and said, okay, well, we really need to quantify this. So do algae release DOC, and how do bacteria subsequently respond to this? So a postdoc of mine, Andy Haas, has been working in Morea, where we incubated several different types of algae in sterilized seawater over a diurnal cycle. We measured the DOC, the change in DOC from control, just ambient water, sterilized filtered water. And we see that different algae have significantly different DOC release rates, with, lo and behold, turf algae actually having the highest release rates, and these are normalized to surface area. And some of the calcified algae and even corals having the lowest DOC release rates. So then we incubated this water with, we inoculated the water with bacterioplankton from the reef environment where these algae were collected with, and measured the change in DOC concentration after bacteria were placed in them. So this is the bacterial carbon yield, which is essentially the drawdown in DOC after the microbes were put into the water. And we see, again, the highest DOC drawdown in the turf algal treatment, and that corresponds to the highest change in oxygen concentration. So as the bacteria are heating up this DOC, they're metabolizing, they're respiring, and they're using this oxygen. So we see different effects for different species, but the turf algae seem to have the largest impact on not only DOC release, but also on microbial communities. So from this work, we've seen that algae can release compounds that seem to be able to cause coral mortality, and that mortality can be prevented with the addition of antibiotics. There seems to be this hypoxia that's created likely by microbial respiration. Some algae have larger effects than others. Some have no effects. Some are benign. Some are more significant. We see different DOC release rates. And there seems to be evidence that it's not just the quantity of DOC that algae or these primary producers are releasing, but that the quality could actually matter too. So not all of these organisms are going to be releasing the same type of photosynthate, and so some of them may be more easily metabolized by the bacteria. So we think that microbes are important components of coral-algal interactions. We obviously still need to really quantify their role in situ, which is much more difficult, but some of Katie Barrett's work for a student has focused on working on that. We need to quantify the importance of microbes relative to other types of coral-algal interactions, and also the interactions that microbes may have with other types of coral-algal interactions. But we know that some of the fleshy turf algae and some species of macroalgae seem to have the largest effects. And we also know that herbivores are really important for regulating these different algal groups. And so herbivores seem to be also, again, now really important for regulating some of these negative coral-algal interactions that could be happening on reefs. So if we review the seminal paper by McCook, again, looking at all the different types of interactions that have been known for corals and algae at the time, 2001, I would just argue that we should include microbial activity on this list. Okay, so now we've seen certain factors affect the composition of reef communities. We've seen that there's a variety of ways by which corals and algae will interact, and we'll hear about many more of them today, but microbes seem to also be important. So I'd like to take what we've learned from these two studies and evaluate the structure of benthic reef communities on islands across the Central Pacific and look for evidence of human impacts. So taking a bit of a historical perspective, and you guys have already seen this figure, historically, in an effort to quantify coral reef degradation, we've monitored changes in coral cover over time. And this is arguably the most straightforward way of measuring is the coral reef doing good or not. Are the corals going away? If we lose the architectural species, the reef engineers, obviously we're going to lose the ecosystem services that these reefs provide. So we've seen evidence of coral declines across the Caribbean. Similar studies have been done in the Pacific. We just focus up at the top here. We see that back in the 1980s, there was a greater number of reefs shown down here that had, shown here, that had higher coral cover than there is now. So these histograms are shifting into a greater proportion of reefs being dominated or having around 20% coral cover. So we've seen coral cover shifting to lower amounts on reefs in the Pacific. So these authors in this paper showed, okay, we're seeing coral cover declining around reefs. So let's go back to that data set and see if the corals are actually being replaced by macroalgae. So in this paper, they looked at coral and macroalgal cover, and they found that since macroalgae weren't actually occupying greater than 50% of the benthos, they assumed that meant that these coral to macroalgal phase shifts have not occurred. And Pete talked about this as well. So they took this as evidence to suggest basically that these phase shifts were not occurring because macroalgae rarely occupied greater than 50% of the benthos. And they went even further to say that this is a lack of evidence that coral reefs are becoming seaweed reefs. And I would argue that that hypothesis wasn't actually tested because seaweed means a lot more than just macroalgae. Seaweed is a generic term that includes turf algae, and actually crustacea, coral, and algae are seaweeds also. So this hypothesis wasn't tested. And several of the authors, or several of the members of this working group who are here today, reanalyzed these data and plotted coral cover and macroalgal cover for the Caribbean, Florida Keys, and the Great Barrier Reef. And they realized, well, hey, if we look at these two functional groups, coral and macroalgae together, they only account for about 19 to 55% of the benthos. So I would argue what occupies the other 81 to 45% of the reef benthos? What is this so-called dark matter on reefs? And can the other members of the reef benthos actually provide insight into the relative health or degradation of coral reefs? Based upon what we know and what I've talked about today, we know the different functional groups of algae. They have different roles on coral reefs. Can we use some of these components to develop what we would expect a healthy reef to be? What ecosystem services do we actually care about when we say is a reef healthy or is a reef degraded? So if we take a look at these different functional groups, remember the coral algae, the turf algae, the macroalgae, we know the CCAs are active rebuilders. They stabilize habitats. Some species facilitate coral larval settlement and survivorship. They grow slowly. They're enhanced by grazers. They release a little DOC. They have limited microbial interactions, have probably many other important roles on reefs. The turf algae are a mixed assemblage of filamentous and many other cyanobacterian algae. Turfs are known to prevent coral recruitment. They grow rapidly. They're opportunistic settlers. They trap sediments, which can also have other impacts. They increase in the absence of herbivory. They release large amounts of DOC. They enhance microbial activity. Many species of turfs probably have allelopathic interactions as well, as you can see many turfs actively growing over live coral. And then the macroalgae are a highly diverse group of larger seaweeds, sometimes calcified, sometimes not, have highly variable roles. Some release DOC, some don't. Some are allelopathic, some aren't. Some are benign. So again, a really diverse group of algae. So I would ask, when we go and consider, is a reef healthy or degraded, are these two things the only thing we care about? Do we only care about coral cover and macroalgal cover? Or do we actually care about what the reef community looks like? Do we care about whether the reefs are still have the capacity to build reefs, to build calcium carbonate skeleton? Are they dominated by the reef builders, the reef building calcifiers of coral, the crustose coral and algae? Or are they dominated by fleshy algae, the turfs and the macroalgae? And I would argue that these two reef ecosystems are quite different and have different ecosystem services. So I was fortunate enough to be able to test some of these hypotheses by collaborating with NOAA's Coral Reef Ecosystem Division, who are mandated to survey all of the coral reefs in the U.S. Pacific every 2 to 3 years. They survey the benthos using benthic photoquadrat techniques, and they've been surveying these reefs since 2002, but hadn't really been able to analyze the data. So through a collaboration with Cameo, or through a Cameo grant, which is jointly funded by NSF and NOAA, my lab has been analyzing all of these benthic photoquadrats. So thousands of benthic photoquadrats have been analyzed. And we've specifically stratified our analysis to the 4 reef habitats at depths between 5 and 15 meters, and all taxa have been identified to the genus level. Currently, the data also include an additional 16 islands that I myself have personally surveyed across the Central Pacific. So right now this includes 5 archipelagos, each which has inhabited and uninhabited islands within the archipelagos, so the Hawaiian Islands, the Northern Marianas, American Samoa, and the Line and Phoenix Islands. And we wanted to simply test the following hypotheses. Is coral cover lower and macroalgal cover higher on inhabited islands? Looking at this as corals decline, do we get macroalgae increasing in abundance? And then we wanted to take this more inclusive, kind of holistic community approach and ask if reef builder abundance is lower and fleshy algal abundance is higher on inhabited islands, to see if there's evidence that we have communities that are either dominated by reef builders or dominated by fleshy algae or these fleshy seaweeds. And then when considering the entire benthos as a whole, to see if there are consistent differences in community structure on islands with and without people. So to look at the data, first looking at coral cover and macroalgal cover, we see variables. So basically we have all of the islands, the uninhabited here, the inhabited in gray, and they're oriented from north to south, so across latitude. And we see coral cover is variable, macroalgal cover variable. There appears to be a habitation effect on coral cover, but actually after you take into account latitude and archipelago, this difference is not significant. And there's also no significant difference in macroalgal cover across inhabited and uninhabited islands. If we look at these two functional groups together, they rarely sum to greater than 50% of the bottom, and there is no correlation, as Pete showed in some of his data. There's no real correlation between coral and macroalgal cover, suggesting that, at least in this static snapshot in time, these two groups do not covary. They're not, as coral declines, you don't see macroalgae increasing. And interestingly, there are several islands, so each of these dots is an island, several islands that are still truly coral dominated, which is exciting to see. Of course, those are islands that are uninhabited. So if we take this broader perspective and look at reef builders versus fleshy algae, we see many of the uninhabited islands showing greater than 50% of the bottom occupied by reef builders, whereas only one of the inhabited islands has the majority of the benthos occupied by reef builders. And we see significant habitation effects for reef builders as well as for fleshy algae. We see a much greater abundance of fleshy algae on the inhabited islands. If you look at the interaction or the relationship between these two, where we have fleshy algal dominance in this quadrant and reef builder dominance down here, we see that islands tend to fall into either one of these categories. The majority of the inhabited islands are up in this fleshy algal dominated quadrant, whereas the inhabited islands have much greater variability. They span the entire spectrum. It's not surprising. Some of these uninhabited islands are brand new, volcanic, actively erupting, volcanic islands. But we see several islands that are within this reef builder dominated quadrant. So just lastly, to look at the entire benthic community as a whole, we see that this is a PCA of all of the different benthic functional groups. These two axes explain over 80% of the variation of data. We see that the main axis, the first component, incorporates turf algae on this side, and the two reef builders, CCA and corals, on this side. And the majority of the inhabited islands sit over here, whereas the uninhabited islands, again, have much more variability, but we have a larger amount of them that are on the calcified end of the spectrum. So this suggests that by taking into account the entire benthos, we can actually learn a lot more about what's happening on these reef communities. So I would argue that we need to perhaps reassess what we mean by reef health. If we care about reef building capacity, we should probably include CCA in our assessments of reef health. We need to basically think more carefully about what we mean when talking about that. From these data, we've seen that inhabited islands tend to be dominated by fleshy filamentous turf and macroalgae, whereas the uninhabited islands have a greater variability, but are more generally dominated by these reef builders. And this study was in no way an attempt to quantify drivers of change. It's just a simple snapshot, and ultimately, in order to be able to determine what's happening, we need to have successional trajectories. We need to have a time component to this analysis, and luckily, my lab just finished analyzing the last time point of the CRD data, so we'll be able to actually add time into this equation. So finally, I hope I've convinced you guys that local human disturbances can determine the organisms that are even present on a given reef community, that microbes should be included in future discussions of coral-algal interactions, and that when considering benthic communities as a whole, greater insight can be gained when considering benthic communities as a whole about the structure and function and ultimate health of these reef communities. So with that, I'd like to thank numerous people who contributed to these data sets and various funding agencies. Thanks.